right, so we're going to pick up right where we left off. And this lecture and the lecture on Tuesday will be trying to finish up innate immunity, as well as a fairly extensive series of lectures today and Tuesday on acquired immunity. So if you guys remember, we left off on this slide here talking about NOD2 and really pulling the story back from innate immunity and how it related to, or how it relates to Crohn's disease. And the relationship is really this protein known as NOD2. And if you remember in the very beginning of that lecture, I alluded to the fact that NOD2 is one of these PRRs, right? Pattern recognition receptors, okay? That help the innate immune system detect endogenous injury, right? And endogenous infection. All right, so NOD2 was first identified as what we would call a susceptibility gene. We mentioned in the beginning that Crohn's disease is not a monogenic disease. There's not one gene mutation that results in the disease. It's polygenic. And so instead what's going to happen is in these types of diseases you have multiple mutations across perhaps multiple genes, right, which in combination lead to the emergence of the disease. So a susceptibility gene would be one of these genes that are identified that promote or lead an individual to be susceptible to the disease. In this case, NOD2 was identified by, in fact, two different investigators. If you look at those dates, that used two different methods and came to the exact same conclusion almost simultaneously. In fact, they were published in the exact same journal issue of the Nature Journal and in back-to-back pages, okay? So, funny how that happens. All right, so the first group did what's known as a linkage analysis. There are different types of analyses that one can do in order to identify how genes relate to disease states. A linkage analysis is one of them, where you have a population that has a high probability or high propensity for a certain disease. You can do what's known as a linkage analysis. And what you're looking for are, during somatogenesis, right, the production of eggs and sperm, you get bulk swapping, what's called homologous recombination and swapping of your genes. That's how we get diversity in our offspring. And so what they're going to do in the linkage analysis is track these big swaths of DNA that are hopping back and forth in offspring in a fairly large population. But a population, the important part about linkage analysis is it's a population of people that are known to have this disease or have offspring that have a high probability of having this disease. And so what they can do is track how, what offspring have the disease and what chunks of DNA they have, and you can develop what's essentially a probability curve. So that's what's here. And what this group here identified was there's a very high probability of someone having mutations in this region of chromosome 16 that they would develop Crohn's disease. And so they developed, or they discovered what would then be called a susceptibility locus, right? So a region, a large swath of genes within a chromosome, okay? Group number 2 took a slightly different approach. Oh, so I will make one note. A linkage analysis sometimes can be confused with what you might read as GWAS, or genome-wide association study. GWAS, if you see that, is when I am not concerned about your phenotype, meaning you don't have to have Crohn's disease. I'm just measuring and sequencing as many DNA sequences, as many genomes as I can, and doing an epigenetic reversal, right? So I'm figuring out from those genes and from a huge population, who are the individuals that have susceptibility to this disease. It's a slightly different process. All right, so the other group took a very strategic approach, and in this case, this is probably the clinical equivalent of the unicorn, right? Where this is a particular family that has two parents that do not suffer from Crohn's disease, but both of their offspring do, right? And so these are one of these anomalies that you say, aha, there's something here. There's a clear genetic link that's likely recessive, right? And so what these folks found was that NOD2 is truncated or shortened, right? So there's some type of mutation that was passed on to their offspring that causes this protein to be shortened in length. Okay, it's still made, but it's shortened in length. And in this case, the truncation leads to what we would call a loss of function mutation, which means the mutation eliminates the functionality of that protein. Okay? And what they found was this, what they call 3020 INSC allele, found right here. It's the shortened version of NOD2. Here's the full length or wild type allele, right? So parent, the father, and the mother. The father has one copy of each. The mother has one copy of each, and unfortunately, they passed each of their bad copies to both of their kids, right? And so as a consequence, their male, their son and daughter, both display Crohn's disease. What really did it for NOD2 being associated and a driver of Crohn's disease was a number of studies that were then published in 2005 in the journal Science. The first was what this one group did was first knock in this mutant allele, this 3020 INSC allele. And what they're going to do is they're going to, at this point in time, we're using embryonic stem cells, so you can use an ES cell. You can introduce a damaged allele, in this case, this mutant sequence of gene, and you can generate a mouse that thinks that this is its wild type, right? So during the production of a mouse line, you can knock in a damaged allele in this case, right? And then they challenged these with a chemical-induced colitis model. So colitis is just infection of the colon. And they found that they got this elevated inflammation and scar formation, which is reminiscent of Crohn's disease. Okay, so I can either express a loss of function truncation of NOD2, or I can go in and just completely knock out NOD2. I can knock out the gene so a mouse line can no longer make it. And when I give them the same chemically induced colitis, you generate similar results. Okay? All right. So how do we treat Crohn's disease? Well, it's a disease of inflammation, right? This is where we've got an issue where NOD2 is lost its function, therefore our inflammation is overly abundant, right, or overly inflamed in our intestines. And so you can give someone corticosteroids to reduce inflammatory gene expression broadly. More specifically, in the past, let's say, decade or so, folks have been using antibody neutralization of this particular cytokine called tumor necrosis factor, or TNF. And this is secreted by macrophages and the adaptive immune system in response to pathogens. In this case, it's being inadvertently expressed, right, as a part of this defect. Okay, so that's about it. I will point out Humira. Most of you guys probably see commercials about them. It is the #1 grossing drug in the entire world. It's this anti-TNF. It's this anti-inflammatory agent where they're basically binding tumor necrosis factor with an antibody and sterically inhibiting its ability to interact with its receptor and trigger downstream signaling and gene transcription. Remicade is actually a knockoff of Humira. It's the 7th most grossing drug. So when you no longer respond to Humira, then the physicians drop you onto Remicade, and then there's another one that's the 3rd most. Yeah, go ahead.
Do they only use Humira for the treatment of Crohn's, or do they use it for other platforms?
No, I'm sure it's got a lot of both what you call off-label uses, but it also has a number of additional anti-inflammatory indications that it's used for. This is the drug Humira that my friend re-engineered and created 13 new patents to get around theirs, which is pretty crazy. One day he'll hopefully invite me to his mansion somewhere. So, all right. All right, so if you're interested in innate immunity and or Crohn's disease, Dr. Steve Cohn, Melody Rukowski are here at UVA and doing some fantastic stuff. Melanie actually has a really interesting project where she actually induces gut dysbiosis. So she hammers your microbiome and then has shown that you actually generate an elevation in the probability to generate breast cancer. And so she's working on dysbiosis and how that might relate to things like cancer progression. Okay. All right, I know that was a little bit of a quick hit. Are there any questions about those last few slides before I move on? Yeah.
So for NOT2, is that a gene on chromosome 16?
Correct. Yes. Yes.
So it's like in that location.
It's in that locus. And so, normally what you would do is find a big locus that has a strong probability of predicting the disease state, and then you would go in and start narrowing in. It happens to be that these guys found the locus, and another group found a specific gene, kind of back to back or somewhat simultaneously. And so it's a nice pairing of papers in that journal. Any other questions? All right. Okay, so we're going to now switch gears and start looking instead more at adaptive immunity. And this is just like innate immunity. It's a ton of terminology. And I wish I could go back and tell these people to name things, happier things, a little more organized things. But as is the case in all of biology, there's just these historic ways in which things were discovered, and those names carry forward even if they came from an area that doesn't really apply to their function. So unfortunately, it's just a lot of vocabulary. I apologize. Just kind of try to slug through it. All right. So we're going to talk about adaptive immunity, that adaptive arm, in the context of what's known as Guillain-Barré syndrome. It's a French, so it's like guillotine. It's not Guillain. Guillain-Barré. And really where I'll put adaptive immunity in the context of evolution. And so really where adaptive immunity came in was where evolutionary tree diverged from this jawless fish, okay? And so the jawless hagfish is the last of this branch of where we branched off, that it only has innate immunity, okay? Now it's alive still today, but about 300 million years ago we diverged evolutionarily. Our predecessors picked up acquired immunity, and this hagfish line that only has innate immunity creates essentially another tree, or another part of the tree. Okay. All right. So whenever we show this, there's always questions about what these slime glands are. Totally disgusting aside is that they actually produce slime. It's a natural defense mechanism of this animal. It looks pretty nasty. I guess they literally slime their attackers to death. But yeah, it should be called the ghost fish, right? All right. Okay. So Guillain-Barré syndrome. It's what we call an acute autoimmune pleuronopathy, polyneuropathy. And so let's kind of break that down a little bit. So we're going to talk a little bit about autoimmune. Autoimmune is when our immune system can no longer recognize self from foreign, or it maybe more specifically, it recognizes self as foreign, okay? So we have a lot of mechanisms in the early development and pruning of our acquired immune system to kill all B cells, all T cells that recognize any self-antigen. Okay, that happens during embryogenesis, but it also actually continues after birth in the perinatal scenario and neonate scenario. Okay, a polyneuropathy, as the name implies, poly, many. neurological, and pathology. So we get many neurological disorders as a function of Guillain-Barré. Okay, and it's the most common form of flaccid paralysis worldwide. So we've talked about this concept of flaccid paralysis. Does anybody remember what disease that was? Botulism. Yeah, excellent. Good memory. All right. Symptoms, pins and needles feelings, nerve pain, obviously severe muscle pain and contractions, especially in the extremities and lower back. Okay. Unsteady walking, difficulty with eye movement, facial movement, obviously any kind of neuromuscular issues. We have autonomic dysfunction, so you have a hard time regulating your heart rate, blood pressure, body temperature, and bladder control. And you have difficulty breathing. Okay? Yeah.
So the pins and needles feeling, is it like that feeling when you're like falls asleep? Is that
Yeah.
Okay.
Yeah. Yeah, when you lay on your arm at night and you wake up and you're like, ah. Yeah. Yeah. It's always fun. Twice we've had, a brand new baby or a small kid who does that, and they don't understand it when they wake up. They're like, my arm, what is going on with my arm? It's always fun. All right. As a parent, right, to watch. All right. Progression. So it's going to follow a pretty straight progression, unlike a lot of neuropathies. We don't understand the progression, of autoimmune diseases. In the case of Guillain-Barré, we have a pretty good indication of how this progresses. So it starts with some type of acute infection that's going to precede all of the follow-on impacts. There's what's known as a progressive phase that exists for about 4 weeks. It's going to plateau, and then the resolution of this disease really takes an exceptionally long amount of time, months to years. And in fact, the statistic is 30% of individuals that suffer from Guillain-Barré still have weakness after about 3 years. It's very difficult to diagnose because, as we just mentioned, it has a lot of sort of non-specific symptoms, right? Shortness of breath, right? Flaccid paralysis. All of these things can be attributed to things like botulism, heavy metal toxin intoxication, polio, tick paralysis. There's just a number of sources of these sort of neuropathic phenomenon and symptoms that one observes in Guillain-Barré. So it's actually quite difficult to make what's known as a differential diagnosis. Yeah, go ahead.
I was just wondering, is there like any specific type of, is there any like root of acute infection that is associated, that's perceived to you guys, or is it like any sort of?
Yeah, it's not as well known. What? Yeah. All right. So it's a bacterial infection. It is most often this bacteria that I'd love to be able to pronounce. Campylobacter jejuni, C. jejuni is how it's usually said. But it can be any bacterial infection. What it's going to induce is an autoimmune attack against Schwann cells. We haven't talked about Schwann cells. Some of you guys are taking physiology, so hopefully maybe you know about this. But in our peripheral nerves, because the signals have to be transduced along the nerve axon, meters sometimes, in a meter in length, that signal needs to be insulated. And so it's insulated by a protein sheath known as myelin. And your Schwann cells are the cells that are making, secreting, and assembling that myelin sheath that essentially acts as this stabilizer of the conduit. Okay? All right. So if we destroy our Schwann cells, we can't make myelin. If we can't make myelin, then we can't insulate those neural signals, and now we lose that connectivity, which is why you can imagine and why many of the phenomenon are sort of peripheral nerve issues, this flaccid paralysis, difficulty breathing, even the autonomic responses, right? There are autonomic nerves that are going from your heart up into your brain in order to regulate your heart rate and things like this. So all of those signals from the nerves to your peripheral systems begin to become affected. All right. Okay, so there are a number of new terms that we want to introduce at this point. The first is already been described to a certain extent, autoimmune disease. And autoimmune diseases are a broad class of diseases where our immune system, usually particularly the acquired immune system, can no longer detect self from foreign. And again, more specifically, it starts to detect certain self-antigens as foreign antigens, okay? All right, so antigen we've described before. It's a substance, can be a protein, oftentimes is a protein, but it can be DNA, can be lipids, can be carbohydrates, right? So any biomolecule or chemical molecule that's capable of inducing an immune response. So that's antigen. If we put these two terms together, you get what's known as an autoantigen. That's one of your proteins or one of your biomolecules, right? DNA, carbohydrates that for some reason your immune system recognizes as an antigen and generates an immune response against. We talked about another one of these. Anyone remember what that disease was? Other autoimmune disease that we've discussed? Lupus. Yeah, that's right. Okay. All right. Epitope, again, we've talked about. So if the antigen is the molecule, the epitope is the specific feature of that molecule that the immune system or that the antibody is going to be raised against. Okay, so it's a discrete part of any antigen that is antigenic, generating an antibody or immunological response. To really expand on this, it's really important that you realize that the antigen is the whole molecule and the epitope is the specific sub-feature, right? So it might be, if it's a protein, it's a peptide sequence or a structure, right? A change in the structure of a specific protein. But the epitope is the feature that is going to be detected. The antigen is the molecule with which it's directed against. And so as a consequence, you can have, and this was, I think we had some discussion about an antibody binding how many antigens. You can certainly have many antibodies raised against a single antigen, right? But each antibody is going to detect a single epitope. Does that make sense? So I can have a protein that has 5 epitopes, right, that are detected by 5 different antibodies. Does that make sense? Okay. All right, more terms. Antibody, we've talked about it in the context of Western blots, ELISAs, biotechnology. In this case, we're going to talk about the origins of the antibody. We've utilized the antibody for these types of approaches, but it originates as a part of our immunological response. And an antibody is a protein that is going to bind tightly to specific epitopes for identification and elimination. And we're going to expand on this concept of what an antibody is. We're going to see it in different forms here in a little bit. Anaphylaxis is a systemic allergic reaction, right? So if I get some people have a localized, let's say, allergic response to a bee sting. Most of us do. But some of us will generate a systemic allergic reaction in response to peanut allergy or bee sting, right? And if it's systemic, then it's called anaphylaxis. All right. Proliferate. We've talked about proliferation, right, and cell doubling. I don't think we've actually defined it explicitly, and so we're going to do that here. Proliferate or to proliferate is when cells are going to divide. So one cell is going to divide into 2 daughter cells. It's going to duplicate its genomic material, right, and generate two daughter cells. And I want to make sure that it's clear this is different than if you hear the terminology cell growth. right? So in the pre-lecture, you heard about the B cell that is going to differentiate and turn into a plasma cell. That process results in cell growth, but that B cell differentiation to a plasma cell is not necessarily going to drive duplication or proliferation. And that difference is really important in immunological response, okay? And that brings us to differentiate. So differentiate or to differentiate or differentiation is when a plastic or progenitor or precursor cell gains a gene transcriptional profile that specifies that cell for a specific function, right? So a lymphocyte, a B cell, right, that turns into a plasma cell, that's a process known as differentiation, okay? Because it's becoming specialized to produce antibody. Okay. All right. So let's get back to these all-important qualities of the immune response, and where in the last lecture we really talked about diversity, specificity, and memory in the context of innate immune. We want to try to focus on these aspects when it comes to acquired immune responses. So diversity, you're actually, without any prior knowledge, you are born with the ability to detect over 1 trillion different epitopes. Over 1 trillion different epitopes that you are, without any prior exposure to a pathogen, capable of detecting. All right? So you're essentially pre-armed with this level of diversity. And we'll talk in a number of lectures from now. We'll talk about how that diversity, that level of diversity, is developed in your body. Okay? Specificity, again, we talked about the ability of antibodies. We'll also talk about T-cell receptors to be highly specific for a single epitope that is presented. Okay? So the epitope can be a single amino acid change, right? So a valine to an alanine, right? Phenylalanine, so you can go on through, right? So it can be a single amino acid substitution. It can be a structural change. So we've actually in the lab raised antibodies against the exact same molecule that has a shift in its conformation of about 1 nanometer. The sequence itself is exactly the same, but we've got an antibody that detects one conformation and not the other. Okay, so those kinds of things are possible. When we talk about memory, in the innate immune system, we were talking about evolutionary timescales of memory, right? These are fundamental pathogenic features, right, like LPS, that are presented generation after generation after generation, right? And as a consequence, we have evolved these sort of standard classic mechanisms to eradicate those kinds of pathogens. In the case of memory of your acquired immune system, what you're going to do is you're going to take, and we'll talk about this later, one of those, let's say 1 trillion, your ability to detect 1 trillion epitopes. We're going to take one of those epitopes, right, that informs a T cell or a B cell, and we're going to then, that epitope will then inspire both proliferation. So now instead of having one cell that detects that epitope, now I have many cells, tens, hundreds of cells that are capable of detecting that epitope and generating an immune response, right? And so memory in the case of acquired immunity is over your lifetime, right? Your memory is related to the pathogens that have infected you. My memory looks very different, right, than yours, okay? All right, so again, the divisions of the immune system. Remember, we've divided them up in numerous ways. One is innate and the other acquired, right? Within both of those branches of immunity, you have a humoral or a systemic, right, blood-driven response and a cell-mediated response. And in adaptive immunity, the humoral response is really going to be centered around the ability of our bodies to make antibodies that bind to and inactivate pathogens. And they'll do it in a number of ways. But that general phenomenon of a B cell turning into a plasma cell, secreting antibodies that are now circulating in our bloodstream, constantly waiting to detect, right, its epitope. That's the humoral response. The cell-mediated response is going to be driven primarily by T cells, and we'll go into that a little bit more. All right. All right, so that brings us to another division, right? So if we look into, if you guys can remember that plot of hematopoiesis, right? The generation of myeloid lineage cells and lymphocytic lineage cells, that's another division, right? Which for the most part, I had a question in the beginning of class, for the most part, the myeloid-derived cells are, in general, the innate immune system. And the lymphocytic cells, right, are primarily acquired immune cells. Right? Now, there's two clear distinctions. One is that natural killer T cell. It's right there in the middle of that plot, if you think about it. And the other is the dendritic cell, which we're going to talk a little bit more about in these lectures, and was presented in the pre-lecture. These are these cells that really are at the boundary, right? They're myeloid cells. The dendritic cell is myeloid derived, but it interfaces very much so with the lymphocytic system. All right, so if we look at those lymphocytes and think about them primarily along the context of acquired immunity, they really fall into two different types. What are known as the B cells, which are going to generate our humoral immunity. And these cells, upon activation, will differentiate into plasma cells that are ultimately responsible for secreting antibodies into the bloodstream. Okay. They will ingest, they can also ingest, digest, and display parts of antigens to T cells. And that's a process known as antigen display or antigen presentation. Okay, so these are professional antigen presenting cells, is what we would call them. Okay. And we'll dig into B cells here in just a minute. The other is that of T cells. They're primarily responsible for cell-mediated immunity. These, in contrast to B cells, will be activated upon direct cell-cell contact. And again, we're going to unpack this a bit more here in the future. All right? They're going to bind to either infected cells or presenting cells. They can deliver toxins. They can produce cytokines. They can produce a number of different immunological responses to the display of that antigen, right? That binding to an antigen, depending on what that antigen is, the level of activation that antigen drives in that cell, and what type of cell it is, right? So there are multiple types of T cells that all have various roles. They will certainly induce apoptosis. I think Kevin talked about this a little bit in the pre-lecture. One of the great things that T cells can do is produce what's called supporins. Right? These are pore-forming proteins that will insert themselves into cells and basically allow the leakage of contents to come in, or allow osmotic pressure, right, to drive the destruction of an infected or cancerous cell. And again, through the secretion, the expression and secretion of cytokines, chemokines, they can modulate the immune system. All right. So let's focus primarily, at least initially, on the B cell side, on humoral immunity. And we're going to focus on these things known as antigen receptors. Right? So both the humoral and cell-mediated responses are going to rely on antigen receptors. In the humoral system, antigen receptors are antibodies. Okay? And there's a wide diversity of how those antibodies are expressed, which is why they're in this broader class known as receptors. All right? An antigen receptor is a receptor whose ligand is an antigen. Immune cells are expressing one and only one antigen receptor. That goes for B cells and T cells. Okay? One B cell, one antigen receptor, it's only ever going to make one antibody. Right? Okay. And this is where the specificity comes into play. All right. So antibodies are one class of antigen receptor that are expressed by B cells or plasma cells. Lymphocyte, regardless of whether it's a B cell or a T cell, lymphocytic activation is going to result when a lymphocyte, either a B cell or a T cell, encounters what's known as its cognate antigen, meaning the receptor is matched to an antigen. Cognate means matching, right? So when my receptor binds its matching or complementary antigen, that's when we'll trigger activation. A little more complicated than that, but we'll talk about that. Okay, so let's talk about humoral immunity. Make sure I'm not missing anything. Oh, just have a note here. When you think about lymphocyte activation, that can result in proliferation, right? So a doubling, expanding of that cell type, whether it's a B cell or a T cell, can actually drive differentiation. So you think about that B cell going to a plasma cell, that can be a consequence of activation. Or, again, in the context of T cells, that can produce cytokines and things of this nature. All right. So antibody, just to remind you, we've gone over this a couple times, but an antibody is a tetrapeptide. There are 2 identical light chains and 2 identical heavy chains. And this is a crystal structure of an immunoglobulin G, or an IgG. And we'll talk about that, the different classes here in a minute. And what is the pseudocolors here are the 2 identical light chains are in this sort of white/gray, and this red here, right? And the 2 identical heavy chains are here in green and in blue. Okay. So what's important is that all of these chains have both portions that would be considered constant, meaning from one B cell to the next, it's the exact same sequence, right? It is unchanged. And both of these chains, both the light and the heavy chain, have regions that are variable, are different between one B cell and the next B cell. Okay? So the variable regions are located in these fragments or these domains known as the Fab domains, the antibody binding domains. And the variable regions are here, right here at the edges of this Y-like structure, right? So this Y structure. The variable domains are here at the tips, on the ends. And both the light chain and the heavy chain are going to have variable sequences in that domain space, in that region of the molecule that help define the specificity of that antibody for its antigen. Within the Fab fragment, each of those chains also has constant regions, right? Regions that are sequences that are the same between one B cell and the next. And that's important, and I'm harping on it because unfortunately this domain down here is called the constant domain, right? So what I don't want you to think is that only the heavy chain has constant domains or constant regions. Both light chain and heavy chains have regions of their structure that are constant, meaning consistent from one cell to the next, and both chains have variable regions, right? Regions that are different from one to the next. I just articulated this. Antigen binding domains are these up here, and the antigen binding face is here on these things called complementarity detection regions. You don't have to worry about that. And the leg of this, in this case, it's an IgG, is called the FC, or the constant region. All right, so if we blow up the antigen binding sites, here's the heavy chain, here's the light chain, and where antigen bindings occurs is on these loops, on these unstructured, so these are Ig or immunoglobulin domain are these antiparallel beta sheets that help hold the protein together with hydrogen bonding, and they have these little loop structures that are connecting these beta sheets. It has 3 of these that that canonically tend to drive antigen specificity. CDRs 1, 2, and 3. Where CDR3 is, again, just, you don't have to know this, but CDR3 is the one that drives the vast majority of specificity and binding to antigen, okay? And so these are the unstructured and variable regions. Okay, great. All right. So because antibodies are multivalent, right, and they can actually bind multiple antigens, right? Not multiple different antigens, many of the same antigen, right? So I want to make sure that we're clear on that, okay? One antibody is going to bind one epitope, but because it's divalent, right? You have these two heavy chains, it has this symmetry. I can bind one antigen on one arm and another antigen, the same antigen, on the other arm, okay? As a consequence, antibodies can actually cross-link proteins. Is this a covalent cross-link or a non-covalent cross-link? Non-covalent, correct. Right? There are no bonds, no permanent bonds that are being formed. Okay? This is all ionic interactions, hydrophobic interactions, right? Hydrogen bonding. All those things can be broken. They can cross-link antigens and form what's known as an immune complex. And so what you can get is multiple antibodies. In this case, let's just, for simplicity's sake, say we have an antigen, and it presents 1, 2, 3, 4, 5, 6 of the same epitope, for whatever reason. Don't ask me why, because we drew a hexagon here, right? And we've raised an antibody that can bind that same epitope along all this protein, right? What can happen is now I can get this macromolecular complex, this immune complex. It can be so powerful that if we do this in, let's say, a tube, I can actually do what's called crashing out a protein. I can form complexes that are so large that they literally fall out of solution and into the bottom of the tube, okay? So this is a process known as immunoprecipitation, where we've generated such a large antibody immune complex that the protein literally comes out of solution. Okay? You'll see immunoprecipitation as a methodology in maybe some of the papers that you're reading. In that case, what we'll do is we'll tether that antibody to a large bead that's really heavy, and we use centrifugal force to pull that bead down after it's bound to its antigen. It's a biotechnological version of immunoprecipitation. The complex formation also facilitates a number of things. So one thing is I can neutralize, let's say, bacteria by clustering bacteria and causing them to be confined, right, through this immunocomplex. But the other thing is that complex formation, as we discussed in the innate immune system, can lead to the activation of the complement system. That's through that FC portion of antibody, right? Will trigger complement cascade. We'll talk about it here in a minute, but there's special features on the antibody that can be recognized by macrophages and neutrophils and can induce those cells to undergo the process of phagocytosis. So whatever that antibody is bound to, they start just engulfing. If an immune complex interacts with mast cells, neutrophils, or eosinophils, that triggering, those multiple antibodies binding to those cells can trigger what's known as degranulation. So we talked a little bit about that in the last lecture as well. Those are granulocytes, right? So eosinophils, neutrophils, mast cells are what are known as granulocytes because they have these large granules of immune modulators, and they're just waiting to dump them out, right? In the case of mast cells, histone is a big one. That's why we get an allergic reaction, or why we get red when we have an allergic reaction is the histone that's in those granules. All right. And I'll just reiterate here that these down here, these 3 bullets on the bottom, this is another place. This is where acquired immunity and innate immunity are going to converge, right? So this is an antibody that is created as a part of the acquired immune system engaging aspects of innate immunity. Yeah?
Do the phagocytes take up the antigens or the antibodies?
Yeah, they'll engulf the whole shebang. They don't care.
Will it like engulf like just a free-floating antibody solution, or does the antibody have to be bound to the antigen?
That's a great question, and I think I'm going to get to it right here. All right, so how do you activate a lymphocyte, right? Or a phagocyte, right? In this case, this cartoon is wrong, Okay, so just make a big wrong. Okay, one antibody binding to one FC receptor. So on the case of macrophages, neutrophils, mast cells, they have what's known as an FC receptor. It is a transmembrane receptor that specifically detects the FC portion of antibody. Well, that's a problem, right? Because I have tons of FC domain rifling around my bloodstream as we speak. Okay? And so I have a ton of circulating monocytes as well that all have, and neutrophils, right? That all have an FC receptor. One FC receptor binding to one soluble antibody is not sufficient to trigger signal propagation. There's some threshold, right, which is associated with the clustering of FC receptors. So remember I said we're going to begin to generate these immunocomplexes, right, of multiple antibodies bound to multiple antigens. And it's in this concentration of a lot of FC domain, right, in a small area that can bind many, many FC receptors, and you get over a threshold, and then you trigger activation of that cell, okay? This is what prevents us, right, macrophages, from engulfing and degrading every single antibody that you make, okay? All right, so the antibodies are going to bind to antigens. They're going to form fairly large complexes. And in the case of macrophages, neutrophils, mast cells, eosinophils, these sort of what we would call non-lymphocytic white blood cells, right? They're going to bind the FC receptors, and once they're activated, they'll cause degranulation or phagocytosis. In the case of mast cells, that massive degranulation is kind of what we associate with things like asthma. So that was a good question. Any other questions about this? Does that make sense? It's like clustering of any other receptor. Oftentimes I can't push a cell in a direction I want it to go, phenotypically, unless I bind many growth factors to that growth factor receptor, right? Or I have to cluster integrin, or I have to, right? So there's almost always some type of critical threshold of receptor binding to ligand, in this case, FC receptor binding to antibody, to get me over some type of threshold to activate the cell. Otherwise, we'd be an activation mess right now, right? Like if every single molecule could activate a cell. All right. So if we talk now about antibody classes, so remember, antibodies are immunoreceptors. They are receptors. And it's a little bit sometimes hard to get our minds around this because we think of them as these soluble proteins that are simply binding epitopes on antigens. And they're receptors because they actually start out as surface expressed or surface tethered molecules. So there are 5 classes of antibodies that we want you guys just to be aware of. And they are always designated by IG and some letter, X, in caps, where IG stands for immunoglobulin. And immunoglobulin comes from this basic structure here, okay? So there is a very large class called the IG superfamily. Many proteins that are not even antibodies are a part of that family because they have this immunoglobulin structure, okay? So IG stands for this structural motif. The ones that you're likely most familiar with that we talk about in Western blotting and ELISAs and these biotechnological assays are IgGs, or immunoglobulin gamma. These are the bulk of the secreted antibodies against bloodborne antigens, and they can comprise as much as 20% of your blood, the proteinaceous component of your blood, of serum. Okay. IgD is an early antigen receptor on B cells. So eventually that B cell will recognize its cognate antigen and will become activated to differentiate into a plasma cell and make an IgG. That receptor, right, the IgD that will eventually turn into a produced IgG, they have the same variable region, right? It is the early or primordial IgG, right, before it really turns into IgG, okay? So it's an early antigen receptor on B cells. IgM, if there's an early predecessor of IgG, there's the first, and IgM is that. IgM is the initial antigen receptor on a B cell surface and is the first class generated in an immune response. Okay. One B cell is going to make one kind of IgM, right? Only one sequence of IgM. The IgM will eventually mature into an IgD, right? And then upon differentiation of that to a plasma cell, it's the same sequence, just slightly different structure, is now turned into a secreted IgG molecule. Yeah? I'm going to show you. Yeah. So it's all structural, right? All right. Let me get through this class. I got 2 more to go, and then I'll get right to the difference between IgM and IgG. IgM is a specialized type of antibody against primarily mucosal antigens. So these are going to be very relevant in epithelial tissues. We're not really going to talk tremendously about them in this class, but it's important that you know because it is a major class. And the last is IgE. So when we think of allergic reactions, it is really the IgEs that are the antibody recognizing those epitopes, right? So they're particularly able to activate mast cells, trigger degranulation, and generate this allergic response. Those are the IgEs. All right, so let's tunnel into these just a little bit, the IgG, IgD, and IgM. And you asked what the differences are. And it is all structural, okay? So here's an IgM molecule. And you can see it's comprised, it's a multimer, right, of many antibodies. Okay? This is what's expressed on the B cell. The sequences that are here, right, at the tips of the Ys, these are the same variable region as the eventual secreted IgG. Okay? So one cell is going to make this initial responder, this initial antibody or immune receptor on its surface. Eventually, the response will be the generation of a plasma cell and the secretion of an IgG, which has a different structure, but is presenting the same specificity to its epitope. Okay? All right. So an antibody thus can be both cell-bound and also secreted, depending on the class of antibody. IgG being secreted, IgM being cell surface, IgD is also cell surface. Okay? In the secreted form, if you remember, it can stick to cells using Fc receptors on those cells, right? Macrophages, neutrophils, mast cells, they have Fc receptors that can bind those secreted antibodies. When they're in the bound state, it's usually because they're synthesized with either some type of hydrophobic transmembrane domain on the end of Fc. And as that B cell matures into a plasma cell, it'll undergo modifications, post-transcriptional modifications. So it'll undergo splicing that we'll talk about later in order to clip those things off so now it can be secreted. Okay? So in the bound state, it has a hydrophobic tail that will be a transmembrane, turn into a transmembrane domain. When an antibody is on a B cell, we actually don't call it an antibody. I know we've been talking about antibodies this, antibodies that. It would be known canonically as a B cell receptor. So when you see a B cell receptor or BCR, that is an antibody that just is simply tethered to the membrane of the B cell. Okay? And as I hope is abundantly clear at this point in time, one B cell makes one variant of a B cell receptor. That B cell receptor is the ultimate antibody that's made, right, in a slightly different form. All right, let me make sure I'm doing okay on time. I am not, so I'm going to speed up a little bit. All right. Okay. So I'm not going to belabor this slide other than to say we've seen it about 5 times now. And we're going to move now to T cells. So we've talked a good bit about B cells, antibodies, antigen. We'll talk a little more about antigen presentation later, but their functions. And now we're going to talk about cell-mediated immunity in more depth. All right. So as we mentioned, on B cells, the antigen receptor is known as the B cell receptor. These are membrane-bound antibodies or membrane-bound immunoglobulins, okay, i.e. antibodies. The major distinction between B cell receptors or membrane-bound immunoglobulins on B cells is that they can bind free antigens, okay? Meaning there's a bacteria that's just hanging around, that B cell can recognize it, right? Or a protein, a bacteria has been chewed up and spit out. The B cell receptor can bind that free-floating antigen, right? That soluble antigen. That's distinguished from T cells and the T cell receptors, okay? So T cells also have antigen receptors that are distinct, although have some structural similarities to antibodies. They're called T cell receptors or TCRs. These are only going to bind peptides, okay? That's a big distinction, right? So an antibody is just binds a feature, and we always use that term pretty broadly. I've seen people that have raised antibodies against polyethylene glycol, of all things, and you can do that in your body, which is unfortunate for the biomaterials community. In contrast, T cells are only going to bind peptide antigens that are specifically displayed and presented by other cells. Okay? All right, so let's talk about the T cell receptors. These two are immunoglobulins in a structural sense. Remember, Ig, immunoglobulin, stands for that structural motif that characterizes antibodies. TCRs have a similar structure. Like antibodies, TCRs are going to bind to very specific epitopes. They're just going to be peptide epitopes. They will exist, they do exist, with an incredible amount of variety in their variable domains. Through very similar mechanisms, right? Your T cell receptor repertoire is equivalent to that of your B cell receptor repertoire or antibody repertoire. Similarly, each T cell makes only one version. One T cell receptor recognizes, and one T cell thereby recognizes one epitope. Okay? But unlike B cell receptors, they are all membrane proteins. They're never made in a secreted form. They're going to be used by T cells to recognize antigens, just like antibodies or B cell receptors are used by B cells. However, they're only going to bind an antigen when presented on another cell, that's not call, by what's known as the major histocompatibility class protein, or MHC complex. Okay? And we'll talk about that. More terminology. MHC protein. And that's cell. All right. So I said they're immunoglobulin, and it's based on this structure. So here's a T cell receptor. T cell receptors are heterodimeric, means they have two chains, right? An alpha and a beta, right? And that's what's in blue and in yellow. Alpha subunit, beta subunit of T cell receptor. It's this structure here and this structure here, these structures that give it its immunoglobulin name, right? So it is in the Ig superfamily. On the left, this is actually the antibody. This is an antibody, an actual IgG, and this is a T cell receptor. All right, so what is this thing so you can imagine based on this structure, right, that the specificity of T cell receptors is similarly going to be out here on the edge, on these unstructured loops, that are spanning between these anti-parallel beta sheets, just like IgG or antibodies are as well. Okay, so this is where the specificity comes in. But I mentioned that T cell receptors can only bind their antigen when it's specifically presented in a particular way. And the complex that's going to do this is known as the major histocompatibility complex. It's also known as HLA, or human leukocyte antigen. This name historically comes from the fact that I will mount an immune response to any part of you that's put into me, right? That is the human leukocyte antigen. You're human, right? And my leukocytes will recognize your human proteins, right? Because they are not self, okay? And they're going to do, so this class of proteins is what really distinguishes you from me, okay? All right. They're going to display, so these proteins are two classes. They're going to display bound bits of protein, so peptide antigens to T cells. There are two classes, as I mentioned. Class 1, these are on all nucleated cells. So every single one of your cells expresses an MH1, a class 1 MHC molecule. Class 2s are going to be expressed specifically on professional antigen-presenting cells. That's those dendritic cells, B cells, right? Macrophages. Those are all professional antigen-presenting cells. I've made a note here. I don't think it's on the slides that you have a link to, but it's here on the recording lecture, and I'll try to remember to put these on here. These are not the same as this blue and yellow. T-cell receptors are heterodimeric proteins. MHC1, this is MHC1 here is in yellow. Here's a critical cofactor known as beta-2, that's supposed to be microglobulin. Apologize for that, microglobulin. And so this is a critical cofactor. So don't think that this is chain 1 and chain 2. MHC class 1 has only got one chain, but it co-binds this beta-2 microglobulin. MHC2 classes are comprised of an alpha and a beta subunit. Okay? Unlike T-cell receptors, they're widely promiscuous, right? They don't care what peptide they're binding. Their goal is to bind any and all peptides and present them to all T cells, as many as they can, okay? So they're not specific to any particular antigen. They'll display any peptide. Here are the two structures. And you can see here these stick figures here and here. This is MHC1 and an MHC2, and this is the peptide motif, or the peptide epitope that it's displaying. It's bound and displaying. They're going to display different sizes, right? So an MHC1 is going to display peptides that are on the order of 8 to 10 amino acids. An MHC2, proteins are going to bind and present peptide sequences that are around 18 to 20. Okay? So about double the size. So the analogy is here is this is like your hot dog with the standard size hot dog bun, right? Fits nicely within the pocket, got a little bit of overhang of the bun. And MHC2 is like when you got a foot-long hot dog in a regular size hot dog bun. All right? So it can hang off a little bit more. Now, the key to remember is that all of your cells have MHC1, and only your antigen-presenting, your professional antigen-presenting cells, or APCs, have this class. All right, go have a hot dog. All right. All right, so why should we care about MHCs? Well, MHCs are the primary antigens that my body is going to use to detect you. Okay? Which is why they're called histocompatibility complex. Okay? Primary antigens that elicit the immune response of a transplant rejection, right? So if you try to give me your kidney, all your kidney cells express your MHC1s. And I can detect the difference between yours and mine. Okay? All right? It gets really complicated. So MHCs are actually polygenic. So we call them class 1 and class 2, but they both come from many, many genes. It's around a dozen different genes that all generate different MHC1s and MHC2s. Okay? So it doesn't come from just one gene. It gets even more complicated. They're also polymorphic, which means they're mutations within them. So even within one gene, I might generate 10 different variants of that one gene, right? Through splice variants, different alleles, genetic variations. Again, tens to hundreds of them. So I've got already a huge variety of these. And to make it even more complicated, they're co-dominant, which means that the ones that you get from your mom interact with the ones that you get from your dad, and they're both important, right? They work together. All right. So it doesn't mean that your mom gives you one set and somehow your dads are recessive. They're both dominant. What it means if I need a transplant is the probability of me finding a match, an MHC class or an HLA match, is exceedingly low. Unless my brother or my sister, not even my mom or dad are really great donors, right? Not even my kids are. It's really siblings are the best donors. It's also why when you work with a transplant surgeon, right, if you have a transplant, you're going to be on massive amounts of immunosuppressors. Massive amounts. Okay. All right. So here's a blow up. I think we're going to be all right. Here's a blow up. Oh, there's a really cool aside here. So there's some evidence, actually multiple reports, that MHC might have an impact on mate selection. Right? So I think so you can imagine if, like, you hand a woman 5 men's shirts, they smell it, right? They're going to pick the one that is the most diverse from themselves. And I suppose this is how we know that we shouldn't be mating with our siblings. I'm not sure. But to diversify, right? Diversify our immunological responses. All right. So here is a crystal structure mock-up of an antigen being presented by, in this case, MHC1. So here's MHC1 in yellow. There's the beta-2 microglobulin in blue. Here's the antigen. That's that peptide motif that's 8 to 10 amino acids long. Hey, I'm from the South. I've got to make the joke, right? All right. And here's TCR receptor. Again, that the variable domain here on the end of the TCR receptor that's recognizing that specific peptide. Right? All right. And so, just like any other receptor system, in the case of TCRs, we need to promote that interaction, right? So we need to make sure that the TCR receptor, because it's binding to a very specific peptide motif, is engaging that peptide motif sufficiently long so that you can generate a response. It's analogous to the FC receptor. I need a certain number of FC receptors to all fire in order to get that cell over a differentiation threshold. In the case of TCR receptors, we're going to use a slightly different mechanism. Yes, you have many TCR receptors that can potentially bind that same peptide motif being displayed by the same antigen-presenting cell, but the probabilities, you can see, start going down as we put limits on how TCR is going to interact with its peptide epitope. So what we do is we've evolved other mechanisms to help stabilize that interaction so that instead of needing 10 TCR receptors to bind, I can get 2 or 3 to bind, but they bind for longer amounts of time in order to trigger a critical signal. And these come through these proteins known as cluster of differentiation proteins, or CD proteins. It's an unfortunate name. It's not in blue. You'll see a lot of proteins, CD90, CD142, we're going to talk about CD4 and CD8. And it just happens to be how they were discovered. So don't pay too much attention to them. They always have other names as well. Thankfully in the case of immunity, they are canonically known as CD4 and CD8. And these are going to describe sort of the two major classes of T cells that have specific kinds of receptors to recognize either MHC1 or MHC2. All right, so CD4 and CD8 determine to what MHC a TCR will bind. Again, whether it's class 1 or class 2. CD4 is going to stabilize contacts between TCRs and MHC2, okay? But not MHC1. CD4 is found mainly on helper T cells, TH cells, helper T cells. And just as an aside, CD4 is the molecule that's harnessed by HIV to infect your immune system. Okay, so you can see there's a little cartoon depictor here. Here's MHC2. It's presenting its epitope. It's going to bind T cell receptor, and CD4 is going to co-bind in this complex in order to help stabilize that interface between T cell receptor, its cognate antigen, and the MHC, in this case, class 2 protein. Okay. CD8 is going to stabilize contacts between TCRs and MHC1, but not MHC2. CD8 cells are found mainly on cytotoxic T cells. Okay. They're also known as T cells, but not so much. Usually cytotoxic T cells. Okay? All right. Here it is, a crystal structure. Here's the TCR receptor on a B cell. Here's that peptide antigen laying right in here in the MHC2 class, right? So you see it's hanging off the ends. So that you know it's MHC2. And in this case, this co-receptor CD4 is going to come down. It actually doesn't interact with TCR. It's going to come down and bind this lower domain on the MHC2 protein, right, to help stabilize that interaction. So MHC2, can't diffuse laterally in the plasma membrane of the antigen-presenting cell, nor can it come apart, right? So it's just to stabilize that interaction. Kevin has a really great way of remembering which CD protein helps facilitate which MHC. I thought it was clever enough that I'm going to present it here as well, which is CD4 times MHC2 equals 8. CD8 times MHC1 equals 8 as well, right? So the sum is always 8, or the product is always 8, right? I actually like it, so I only have to remember one number, it's 8. And now, if you tell me the number is 8, and I ask you which CD protein binds MHC2, you know immediately it's 4, CD4, okay? Nice, clever little way. All right, so we're out of time. We'll pick up right here on Tuesday. There is a quiz on Tuesday on all the pre-lecture and lecture material.